Welcome back, my dear students. In this presentation, I am going to explain about the principles of inheritance and variation part 2, that is Mendel's laws. The first law proposed by Mendel was the law of dominance. According to law of dominance, the characters are controlled by discrete units called factors. Whatever the character may be, it may be a tallness or maybe the dwarfness. So these tallness or dwarfness character is controlled by a unit called factors, and according to him the factors occurs in pairs. That is, the factors always occurs in pair, and in case of dissimilar pair of factor, so one dominates the other. Dissimilar means heterozygous condition, for example T and small t. So here the capital T dominates over the small t. See, this is the plant. Here this is tall plant and it is represented by capital T and capital T. And here this is dwarf plant which is represented by small t and small t. Okay, but if in this case capital T and small t, always the capital T expresses whereas the small t will be suppressed. So the character will be always tall. So this is called the law of dominance. Inheritance of one gene, it is also known as law of segregation, purity of gametes, or monohybrid cross. In the last session only I have told you mono means the cross which involves the single character, right? So according to this law states that when the alleles for two contrasting characters are brought, see, these are the parents, that is pure tall and dwarf. The phenotype of these parents can be written as capital T, and for dwarfness we can write as small t. When it produces gametes, so gametes should be encircled like this. The T will be taken from this and small t will be taken from this. And syngamy, it is also known as fertilization. In the F1 generation, all plants are found to be tall because of law of dominance. Here one capital is there and one small is there, means the capital character always express because dominant gene always express. So all F1 plants are found to be tall. Next, when the F1 are self-crossed, that means an F1 is crossed with the Fn, then what happens? The capital T small t and capital T small t is crossed. When it produces gametes, it produces two kinds of gametes. One is capital T gamete and second one is small t. Here also the same thing, that is capital T and the small t. And we have to represent it in the form of a Punnett square. Okay, so this shows the nature of F2 generation. See, during the Punnett square, we have to draw like this. And finally, capital T small t and capital T small t. So you have to multiply this capital T with capital T. That comes the homozygous tall. And again capital T with small t, that is heterozygous tall. Again small t with capital T, but you have to write capital T then small t because law of dominance. So this comes the heterozygous tall, and next is small t, that is homozygous dwarf. Then you have to count the genotypic ratio and phenotypic ratio. The genotypic ratio is found to be 1:2:1. That is 1 homozygous tall, 2 heterozygous tall and 1 homozygous dwarf. And the phenotypic ratio is found to be 3 tall plants and 1 dwarf plants. So this is known as inheritance of one gene. And the law states that when the alleles for two contrasting character, that is pure tall and dwarf for the two contrasting character, when they are brought together in F1 generation, see here this is the F1 generation. Okay, they separate. Yes, they are separating here. And finally, they will segregate in F2 generation during the formation of gametes. So this is known as inheritance of one gene, law of segregation or law of purity of gametes or monohybrid cross. Fine. Fine students. Now let's go for the test cross. The test cross is said to be the cross between the F1 hybrid and the double recessive parent. Here the F1 hybrid is capital T and small t, right? See, heterozygous. And here double recessive parent means small t, small t. When they are allowed to produce the gamete, here they produce capital T and small t as gametes. And here the capital T is not there, but small t and small t is produced. Now, the result what we get is the 50% of them is tall and the 50% of them is dwarf. So, or else we can also say it as 1:1 ratio. So, this is known as test cross. Then what is the significance of doing test cross? First, the significance is it is done to confirm that F1 hybrids is homozygous or heterozygous. Second significance is it helps in knowing the genotype of an individual. Okay, the genotype for a particular character, whether it is tall, whether it is dwarf. So what is the genotype of tall? In order to know this test cross is done. And finally, this helps in predicting the genotype of test organism, what test organism we have taken. So these are the three significance of test cross. Now, let us go for the inheritance of two genes. It is also known as the law of independent assortment or dihybrid cross. Dihybrid cross means it is a cross which involves the two characters. Here, the two characters which are taken by the Mendel was round yellow and wrinkled green. Round yellow can be written as in the genotype capital R and capital Y, and for the wrinkled green small r and small y. And during the production of the gametes, this can be written as capital R Y and this can be written as small y. And during the syngamy or fertilization, it produces capital R and capital Y small y. And from the F1 generation, we found out that all F1 hybrids are round yellow because of law of dominance. See here, capital R is there means it is round. Okay, and capital R is there means it is also round because of law of dominance. Next, small r means it is wrinkled. Again, capital Y is yellow and capital Y small y is also yellow because of law of dominance, and small y can be taken as green. Now, if F1 are self-crossed, the genotype would be capital R, capital Y, small y, and similarly here also the same. And to write the gametes, put a small checker box here or a Punnett square. See here, capital R and small r are taken in this side and capital Y and small y are taken in this. So you just crisscross it, you will get the gametes like this. And now we'll go for the Punnett square to show the nature of second filial generation or F2 generation. See here, the same you have to write capital RY here and capital RY this here, and finally you have to write the same thing here and here, and you just crisscross it and multiply it, you will get capital R, capital Y, that is round yellow. Again capital R, capital Y, small y, again round yellow because of law of dominance. Similarly, you just do the same thing for each and everything. This row you will get again round yellow. This row you multiply capital R like this, and capital Y and small y, because here you have to write the capital first, law of dominance. Again same you do with the second one, capital R, small r, y, like this you just do for everything. You will get the round yellow 9 and round green 3, wrinkle yellow 3 and wrinkle green 1. So the phenotypic ratio was found to be 9:3:3:1, and the genotypic ratio is found to be 1:1:2:4:2 and 1:1. So this is called inheritance of two genes or law of independent assortment. Fine. Now we'll go for the test cross for the dihybrid cross. The test cross means the cross between the F1 with the double recessive parent. Here the F1 is capital R small r and capital Y small y, and here the double recessive parent is small r and small y. So you have to write like this, capital R Y and capital R Y. For this I am showing, and here the gametes are small r and small y. You just multiply it, you will get, that is while multiplying the, this one is this one, capital R small r, small y, like this, and for multiplying this and for multiplying this and for multiplying this, you just multiply 4 and you'll get the 4 possible results, and this shows the genotypes, and the phenotype would be the round yellow for this one and round green for this one and wrinkle yellow for this one and wrinkle green for this one. So the test cross ratio was found to be 1:1:1:1:1. So this is the test cross ratio in case of the dihybrid cross. Right. Now let us move towards one more concept which Mendel failed to explain. That is the deviation from Mendelian principle. This is also known as incomplete dominance or partial dominance or blending inheritance. And this can be seen in a flower color in Snapdragon. So flower color in Snapdragon can be given as an example, or else 4 o'clock plant. So these are some of the examples for incomplete dominance. Incomplete dominance says that when the two heterozygous alleles are together, one of the dominant allele failed to express. Okay, and here what happens? As a result, both the alleles together express an intermediate character. Let me explain you. That is, here the parents are the homozygous red flower plant, and red can be taken as capital R and capital R. And homozygous white flowered plant, we can write it as small r and small r. So this produces a gametes, and we can write it as capital R, and this we can write it as small r. And during the syngamy, the F1 are found to be the pink. Actually, capital R means red, small r means white. According to law of dominance, red should come. But here it is showing what? Intermediate character. Here it is showing the intermediate character. So this is the deviation from Mendelian principle or the incomplete dominance. Now let's move further. When this F1 hybrids are self-crossed, capital R and small r, again capital R and small r. So they produce two kinds of the gametes again, and we have to write the Punnett square for the second filial generation, capital R and small r. Here similarly capital R and small r. See here it produces the red flowers, and here capital R and small r means it is pink, intermediate. You have to write pink here. Again capital R and small r, here again pink, and here it is expressing the white. Now let's go for the phenotypic and the genotypic ratio. The phenotypic ratio is found to be the red, pink and white in the ratio 1:2:1. Similarly, the genotypic ratio is also 1:2:1. One for the capital R and capital R, which represent the red flower. Okay, one is the capital R and small r, which represent the pink flower, which comes in the ratio 2. And the last one is the small r and small r, that is in the ratio 1, which represents the white flower. So this is the phenotypic ratio and the genotypic ratio: 1 red, 2 pink, and 1 white, and 1 red, 2 pink, and 1 white. Okay, so this is about the deviations from the Mendelian principle or the incomplete dominance. Okay, so these are the types of the plants which are developed, that is red, pink, and white. Hope you understood. Thank you for listening.